I want to put the part that relates to innate immunity up here. So there were two parts to innate immunity. You guys remember what the two sections were that we divided it into on the front side? Should be internal and external. And so I'm just going to put those here. I'm going to do external first because I want to write a little bit about internal. External and internal. And the part that relates to the acquired immunity that we're going to talk about today and kind of relate with the two together is with the phagocytic cells. So I'm going to write here phagocytic cells. Can somebody remind me what phagocytic cells do? What do phagocytic cells do? That's correct. All right, so they go through this process of phagocytosis. Remember I drew something like this where it indents around it and brings in the bacteria or virus or whatever it is, and then eventually the lysosome binds with this vacuole. The lysosome contains enzymes that digest it, and then eventually the cell presents, so I'm going to put here, like this is their protein on the membrane here, presents the antigen here. And so these guys here, I'm going to put here, we call these antigen-presenting cells, or APCs, but you might want to write out antigen-presenting cell, if you're not going to remember that. And there are different kinds of antigen-presenting cells. I'll pull up some pictures here as we talk about it here. As we go through, I'll relate these to some of the diagrams that we talked about in the notes. So that's what we have here. Here's an example of an APC would be, in this case here, this is a dendritic cell. It's engulfing the bacteria, so I'll put in parentheses here, dendritic. Or macrophages do this as well. And so what I'm drawing here is this figure where the bacterium is taken in. Here are the antigens, and it is presenting the antigen, hence it's an antigen-presenting cell. And so, so therefore, this is how the macrophage is going to relate to the acquired immune system. So let's look at the acquired immune system. So I'll come back to this picture in just a second. Just turn that off again here. So let's look at our acquired immune system. There are two sections of our acquired immune system, two categories. So tell me what they are. One involves the B cell. What do we call the part that involves activating a B cell in immunity? Starts with an H. Humoral. So, yeah, here. So I'm going to put in parentheses, this is involving B cells. And then the other part of our acquired immune system is called something else. Cell mediated. And these involve, not the B cells, anybody remember what kind of cells this involves? T cells, and specifically a specific type of T cell. Cytotoxic T cell. Make sure we can see that. All right, yep. Okay. A cytotoxic T cell. And so, these two sections have different purposes in fighting your infection of your pathogen or the rhinovirus, like the video that we just talked about. What kind of cell helps to stimulate your B cells and your cytotoxic T cells? They're not, these types of cells are not really put into either one of those categories of immunity, the humoral or the cell-mediated, but they're very helpful in immunity. That's my clue. They're helper T cells. They're helpful in immunity. They're helper T cells. All right, so helper T cells are in the center here. So I'm going to put here. And they're going to help stimulate B cells and stimulate cytotoxic T cells so that they can do their jobs in fighting off an infection. So let's do B cells first. So actually, let's talk about helper T cells first and then how they stimulate B cells. Helper T cells. This is how this part here that we just talked about with innate immunity relates to acquired immunity. Because this process right here is what triggers and stimulates the activation of helper T cells so that they can help both of these things on these parts of the immunity. So in this particular picture here was the phagocytosis, and it was presenting. Here's the antigen that the dendritic cell is presenting. So that's right here. Notice here what happens is a specific helper T cell. Why is it specific? Because there are different kinds of helper T cells, just like there are different kinds of B cells. What makes them different is the receptors on the outside. Notice that this helper T cell has a receptor with a complementary shape that matches the antigen that phagocytic cell is presenting. So these helper T cells bind together. Now, the molecules, the MHC molecules and the CD4, this is detail. In fact, the video did a lot of detail about CD4, CD8, MHC1, MHC2, and so on. For the AP test and for our class, I'm not going to ask you specifically about does a helper T cell have a CD4 or a CD8? I'm not going to, you don't have to memorize those names. We just need to understand the big picture here. So therefore, these guys have receptors that bind, this helper T cell binds to this particular antigen. And so therefore, that binding stimulates the helper T cell. So let's go ahead and on our big picture here, let's draw that here. So, so now I'm going to go ahead and put an arrow over here to show how this relates to this. So this is going to stimulate your helper T cell. So here is, I'll call this I'll just put APC for an antigen-presenting cell. And so let's draw that here. So here's your antigen-presenting cell presenting the antigen. The antigen in this case here is a little triangle in my case here. And so it's saying, hey, we got an infection here. This is what the little guy, the antigen, looks like, this triangle here. So then the correct helper T cell binds to it. And so the correct helper T cell has the correct receptor that matches it. So this is a helper T cell. So we have the helper T cell stimulated here. So remember, this is a form of cell communication when two cells bind together like this. So this binding here triggers a signal transduction pathway that's going to lead to a response in this helper T cell. It's a connection with the cell signaling chapter there. What's the response that's going to happen inside the helper T cell? The helper T cell is going to make and release what? these molecules called cytokines. And so that's your response, is the cytokines being released. So here we have, all right, the release of cytokines. I'll put in blue here. So what do the cytokines do? They are a messenger, and they communicate with 3 different kinds of cells. So this helper T cell is now activated by binding to the antigen-presenting cell. It releases the cytokines. What's one type of cell that these cytokines are going to communicate with? Yeah, it's going to communicate with B cells. So I'm going to go ahead and draw from here to here that these cytokines are going to come over here and stimulate the big B cell. Or they can communicate and stimulate T cells, right? So here we go. All right, so the cytokines. So I'm putting all the cytokine stuff in blue here. Or what's the third type of cell it can communicate with? The cytokines. Yeah, the cytokines can help communicate with the helper T cell as well. And so I'm going to go ahead and draw here like this. Communicates with the helper T cell. And what happens is when it communicates with the helper T cell. So when it communicates with these guys here, here's the receptor molecule. The cytokines bind here. And what usually happens when a signal molecule binds to a cell? It signals a signal transduction pathway, and you get yet another response. Does anybody tell me what the response for this cytokine binding is? What does this helper T cell do? It's not really on this picture here. It clones itself, absolutely. And so this is the self-communication here. It's communicating with itself. And in this case here, you're going to get yet another response. So this is a different response than the release of the cytokines. So what that response is these helper T cells. So this response right here is this right here. It clones itself, and then it differentiates into 2 types of cells, the helper T cell. One that can be used later, so we call those memory. So we make more memory helper T cells. That way, if we ever come into contact with that pathogen later, you have memory helper T cells that can immediately start secreting cytokines and stimulate humoral and the cytotoxic T cells as well. The second type of cell is it divides and clones itself and makes a lot of what we call active helper T cells. What are active helper T cells going to do? These guys are going to be just like this cell. They're going to secrete cytokines. And the cytokines are going to stimulate more B cells and more cytotoxic T cells. So that's the whole purpose of the helper T cells, is to release these cytokines to cause these B cells and cytotoxic T cells to become active. So that's why they're kind of in the middle. They're helping. They're not really involved in either one of these. Notice that the helper T cells, they're not doing anything directly to help with the infection other than to support these 2 other types of cells. All right, so let's look at the B cells first then. These cytokines, how do they stimulate the B cell? Let me go to another picture that was in our notes to relate it to this. have here. This part of the picture right here is what we just drew here. So the antigen-presenting molecule, in this particular case, it's a macrophage. So remember, it could be a dendritic cell or macrophage. So it's presenting the antigen, helper T cell binds to it. Helper T cell goes through this process, releases the cytokines, makes active helper T cells. So that's what we have here, an activated helper T cell. So what is it going to do, this activated helper T cell? Is it's going to stimulate a B cell. Now the B cell, remember that B cells go through clonal selection. So before we even look at any of this here, your B cell has these receptors that the bacteria, in this case here, it's a bacteria, can bind to and have the appropriate shape to bind with the antigen for that particular bacteria. Some of that bacteria goes into the B cell breaks it up, and it presents the antigen. So that's what we have here. And so the selection process in this picture has already occurred. We have hundreds of different B cells. Only this one has the right shape to bind with that particular bacteria, so this is the one that needs to become activated. So how does the helper T cell know which ones are the right B cells to activate? It's because the B cell presents this antigen here. So that's what I'm going to show you here. So when we look at the B cell, so we get here B cell, and this is the right B cell because it has a receptor on it and it's presenting the antigen. You know what? Let's do it like this. Let me keep it consistent like I drew it over here. So this right here, presenting antigen. So then, now this active helper T cell, the active helper T cell that's releasing the cytokines is going to activate this B cell, and it does that in two different ways. By binding to it, so notice here the activated helper T cell binds to the B cell as well as secretes cytokines. So when we look at this active helper T cell, let me put a receptor on it. So that looks like this. Notice that this activated helper T cell has a receptor that will fit into the antigen here. So let's go ahead and draw that here. So here's your helper T cell. They can fit together perfectly. So these cytokines right here, we can say that these cytokines are binding to the B cell. Blue here, binding to the B cell. And realistically, I drew here from the helper T cell. It's really going to be coming from this helper T cell. All right, the cytokines. So I just drew it over.
Would that come down from, would that line come down from the active helper T cells, or would that come down from those cytokines?
Would the line come down? What do you mean?
Like the line that you just drew to the B cell? Would
This
that,
one?
Yeah. Would that come down, would that come from the active helper T cells?
Yes, 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 absolutely. And I didn't want to, I guess like we could draw an arrow if you want to. I didn't want to make it too busy with too many arrows.
I was just wondering.
Yeah, yes. So, so this right here would be this right here, all right? So two things activate the B cell. The first thing is, number one, is the binding of the cytokines. Can activate the B cell. The second thing is right here, the binding of the helper T cells. So those two things, this B cell says, okay, we're activated now. An activated B cell, what that does is now it triggers it to do what divide and differentiate. So that's what you're seeing here in this particular picture here, is that it divides and makes clones of itself, but in differentiates and makes plasma and memory cells. So that's what I want to talk about right next, is two things here. So you're going to have memory cells. And then we have active plasma cells. Remember, memory cells are always for later use, for your second infection, a secondary infection. Your active cells are always for fighting your current infection. So active plasma cells, what do plasma cells do to help fight the infection in the body? They make and secrete antibodies. All right, so plasma cells here, these guys, make and secrete antibodies. And remember that antibodies have a particular shape that will fit, they're kind of Y-shaped like this. That's what they look like. And they have shapes that will fit the antigen on the surface of your pathogen. And so when we say we make and secrete antibodies, remember, does anybody remember what the antibodies' role in infections are? So why is it that so important that we activate these plasma cells and secrete these antibodies? What do the antibodies do? Remember? So if we go to, get another picture here. Right here. So the antibodies can bind to the pathogen themselves. So remember this picture from the notes that they bind to the pathogen themselves. And we talked about these 3 different ways, but all of these guys enhance and make it easier for the macrophages to ingest the bacteria. And so they stimulate or help or enhance phagocytosis. And so making and secreting antibodies, I'm going to go ahead and connect it back over to our innate immunity and write the word enhances. This enhances phagocytosis. And so really, all bacteria and all viruses are gotten rid of through the phagocytosis process, but that process is helped and is more efficient with the use of antibodies. And that's what that picture was showing. So in this humoral or B cell part of the immune system, if we were to say the big picture as far as what this part of the immune system does, what does this part of the immune system do? What does it attack, we would say? We're ultimately fighting directly against the pathogen itself, right? Because the B cells, even though do any of the B cells actually do anything to the pathogen to get? They don't. They just secrete the antibodies, but the antibodies bind to it so that your phagocytic cells can take in the pathogen easier and get rid of them that way. And so, so therefore, I'm going to put in red here, kind of to stick out here, the big picture of the humoral immunity. We could say it enhances the attack on the pathogens. So this is helping to get rid of the pathogen themselves, kill off the pathogen themselves. I put enhances the attack on pathogens. Why? Because the B cell themselves do not kill the pathogen, but the B cell's behavior by secreting antibodies helps with the getting rid of the pathogen. Does that make sense? Okay? All right. And that is the humoral or B cells, the MVP. So then we have one part left here, the cell-mediated or cytotoxic T cells here. And the cytotoxic T cells are also stimulated and activated by the helper T cells and the cytokines. So let's go to cells, this picture here. Guess I don't have my pictures. Alrighty, so let's look at this picture, I guess. I thought I had a different picture here. Okay, so your cytotoxic T cells. So looking at this picture here they are, alright? The cytotoxic T cells also have a receptor. So there's lots of different cytotoxic T cells that we have. Only one will fit the shape of that particular antigen. And so this is your target cell. This is an infected cell. And the infected cell, what they do is, as they become infected, they also display the antigen. And so let's look at here. So let's draw our cytotoxic T cell. And the cytotoxic T cell has a particular receptor that has the right shape to bind with the antigen of that particular pathogen. And this cytotoxic T cell becomes activated when, what happens? So here's your cytotoxic T cell. What would, this picture is showing you it binding to an infected cell. But how it becomes activated to even do that is with the help of the helper T cell and the cytokines. And so we can, I'm going to draw this arrow here from the cytokines from the helper T cell. You actually have a receptor protein on the cytotoxic T cells, and here's the cytokine binding to it, so my blue cytokine. It's going to cause a signal transduction pathway, and you're going to get a response to it happen. In this case, what do you think the response is of the cytotoxic T cell? Look at the picture here. What does it do, the cytotoxic T cell, according to this picture here?
I guess that it would make perforated enzymes.
That's right. It makes and releases them, the perforin and the granzymes here, which are just enzymes. And so, so the response here is to release perforin. The second thing that has to happen is that the infected cell binds to here. So here's your infected cell. Just put your infected cell. It's displaying the antigen of the pathogen that infected it. So I should say that those two things kind of in conjunction trigger your response for the perforin to be released. What does perforin do? Let's remember. Perforin's the one, it pokes holes or makes pores in the infected cell's membrane. So this perforin here is going to make holes or pores, all right, bunch of pores in here. So I'll put pokes holes. It's a little bit more in depth than that, but in the membrane. what that does is allow the second thing that's released is, I'll just put enzymes here. The enzymes can go in as well. And both of these things, the enzymes and the perforin, cause what to happen inside the cell? Apoptosis. Alright, so therefore, the enzymes, I'm going to put these two things, green enzymes and the perforin, those two together trigger apoptosis and the cell dies. Yeah, so excited to put this in here. I forgot to say that one of the responses that triggers this to happen is that the same thing happens over here. I forgot to put here that the cytotoxic T cells actually do make memory cells as well, and active cells, just like the rest of them. And it's the active cells that, when this cell divides and differentiates and makes more active cytotoxic T cells, these guys can go and find more infected cells and fight the current infection. So these guys go in and find the infected cells. How does they know it's infected? Because the infected cells display the antigen. And so this process is happening a lot because this cytotoxic T cell divided and makes many of them. So what could we write in red about the cell-mediated response? What's the big picture here? What's the attack on here? Yeah, this attacks infected cells. So these are actually human cells that have been infected, and so we don't want those human cells that have been infected to be able to, if it's a virus, viruses take and are reproduced in infected cells and can go out and infect other cells. We don't want that infected cell to divide and make more infected cells, and so on. So this gets rid of all the human body cells that have been infected, and then the B cells aid in getting rid of the pathogen itself, so that by the time these two things are going on simultaneously, you get rid of the pathogen, you get rid of your infected cells very soon after your attack on your body is no longer, because there's nothing there left to cause sickness or whatever it is. All right, and that is the immune system. All right, the acquired part of the immune system and how it relates to the innate. Is that helpful? Okay. All right. All right, so this will stop.